Hi everyone, thank you for joining our video on Drug Repurposing Project in Oncology. Drug repurposing is becoming an increasingly important strategy in biomedical research. In this video, we will explore what happens when you take a class of medicines that has been used for more than a half a century to treat diabetes, for example, and ask if they might have something to offer to cancer patients too. I'm joined today by Dr. Mateusz Tomczyk from Silesian University of Technology. We will talk about what first inspired this line of research, why drug repurposing matters as a strategy in oncology, what makes these combinations particularly promising, and what it would take to move a finding like this from the lab to clinic. Mateusz, thank you so much for allocating your time to join our call. Please tell us a few words about yourself and your research focus. The floor is yours.
So hi everyone. Thank you, Irina, for involving me here. I'm very happy to be here. The research we talk about today, I started years ago as a PhD student. Now I have the possibility to lead the grant by Foundation for Polish Science, in which I learn what are the expectations of market, and I can use this knowledge to transfer some scientific results into the development project.
So let's deep dive into your drug repurposing project concept. So sulfonylureas have been used for diabetes for more than a half a century. But what made you look at these old drugs from a completely different perspectives as potential partners for cancer therapy?
So basically, we discovered this idea by accident. I was studying a totally different topic. That time I was looking for inhibitors for hexokinase second, this kind of very important enzyme for cancer cells. And during the virtual screening, we identify many potential inhibitors for this enzyme. And then when we test some of them, we actually found that they are not working well with the hexokinase. Very different project that time, but we found that once we combine sulfonylureas with anti-cancer drugs, the response from the cancer cells was much stronger. And then we start digging in this topic, and we found that sulfonylureas can have additional action. They have very long clinical history in diabetes, but they can be also used as a potential modulators in cancer therapy. It is because apart from their very low action of ATP-sensitive potassium channels, there are many evidence that some members of this family can also interact with other cellular systems, including very important transport proteins that are highly relevant in cancer. That discovery immediately raised an interesting possibility. Let's say that one of the major problems in chemotherapy is that cancer cells can actively remove drugs from themselves. Doxorubicin is a very good example here. It is very effective anti-cancer drug, but resistance can develop, for example, through ATP binding cassette transporters, such as P-glycoprotein. And sulfonylureas can be used to inhibit those transporters and increase the concentration of anti-cancer drugs inside the cells.
You recently published a comprehensive review covering almost 70 years of research of sulfonylureas. what was the most important conclusion from looking at the entire history of this drug class?
I think it was that we should stop thinking about this class of drugs as if they were one pharmacological entity. When we mapped the literature drug by drug, we found very different profiles of those drugs. For example, glibenclamide has considerable evidence related to drug transporters and hemosensitization, while gliclazide, for example, has much stronger evidence around antioxidant, anti-inflammatory, and DNA protective effects. So the question is not really, do sulfonylureas have anti-cancer properties? Because it is too broad. Same therapeutic class may have very different secondary pharmacology, and sometimes that secondary pharmacology can become new therapeutically useful one.
So when you moved from this general idea to experiments, was there one drug combination that clearly stood out?
Yes. We tested many commercially available sulfonylureas in combination with different anti-cancer drugs across several cancer cell models. And we found that doxorubicin with glympyrite give particularly striking effect in breast cancer cells. This combination gives approximately 4 to 5-fold improvement in cytotoxicity compared to just doxorubicin. And then we moved into in vivo models. We use 41 breast cancer model, and we confirmed that these results can be preserved in vivo models.
Considering all these experiments that you have already performed, do you already know why glimepiride makes doxorubicin more effective?
So one of the most important observations from our experiment was that in several cancer cells, the presence of sulfonylurea, for example, glympyrite, increased the intracellular concentration of doxorubicin. And that is exactly what you might expect if the cells becomes less effective at pumping doxorubicin out. It is probably because ABC transporters such as P-glycoprotein or related proteins are plausible candidate for these drugs. Multi-drug resistance is one of our leading hypotheses. One of the observations from our experiment was that in several cancer cell lines, the presence of sulfonylurea, for example, glympyrite, increased the intracellular concentration of doxorubicin. And this is exactly what you might expect if the cells becomes less effective at pumping doxorubicin out.
So your newer work suggests that the formulation of drugs can completely change the behavior of the combination. So why is formulation so important?
So this is probably one of the most important tasks in our recent work. At first, it is very tempting to think about drug combination simply as a drug A plus drug B. But inside living organisms, those two drugs do not necessarily arrive in the tumor at the same time, at the same concentration, or remain in the cells for the same period. So we compared few doxorubicin formulations, like free doxorubicin or nanoparticle doxorubicin, with different combination of sulfonylureas. And we found that once we use liposomal doxorubicin, the effect of the combination can be even greater. This is probably because those two drugs have very different pharmacokinetics. So, but by somehow aligning them together, we can increase their synergy.
If these findings are confirmed in future preclinical and clinical studies, which we hope, how could they influence the treatment of patients? Could these combinations impact different therapeutic areas in oncology?
I believe so, because many cancers share exactly the same molecular basis for multi-drug resistance. So once you found something that works in breast cancers, it is highly possible it will also work in another model. And the very important thing we learned from our current project, from the Foundation for Polish Science, is that we can make the highest impact in the area where big pharma are not present. It could be, for example, kind of rare cancer, especially those in which doxorubicin is still the main therapeutic options. And yeah, there is many disease like this where the medical need is still very high.
Absolutely unmet. Mm-hmm. So, as an innovator, I would like to get your opinion on importance of early engagement with the regulators and interactions through regulatory science tools such as orphan drug designation, breakthrough designation, or innovation task force for the drug repurposing innovation.
Yeah, and that was one of the most important lessons for me as a young scientist, because we want to develop something and then introduce it into market, because this is probably the best way how we can reach people. And of course, we don't want to waste our time to make data that later will be not recognized by the regulators. So it is very good to start a scientific project not only from the perspective of basic science, but simultaneously take care about regulatory aspects. And sometimes it is even more helpful because many programs already have documents that can lead you through the whole project. They list specific results they accept from us, so we can use it to make our projects better.
Fantastic. So, if we look 5 or 10 years ahead, what would have to happen before a combination like this could realistically reach patients?
So there is still several steps to make. First, we need stronger biological validation, and that probably means additional tumor models, particularly models with well-characterized multi-drug resistance and very strong confirmation of the mechanism. And second, we also need more. Pharmacokinetic data. Of course, we want to know not only whether the tumors become smaller, but we also want to know how much doxorubicin or glypyrid actually reach the tumor. And other important health organs. And third thing is that I think pharmacokinetics will be central. If we combine two or few drugs, it is very important to deliver them together in synchronized way so they can express their simultaneous. So, okay, and third, I think pharmacokinetic study will be very important because if you combine two or more drugs, it is very important to deliver them together. So their synergistic action can be accessed in cancer cells. And of course, safety. As we know, many anti-cancer drug like doxorubicin has well-known toxicity. So if we combine drugs together, we should be very careful about the effect they can exert together. So any combination has to demonstrate the improvement in anti-cancer efficiency, but also they should be less toxic, or at least the same toxic as anti-cancer drug alone. And finally, translation will require more regulatory expertise and probably intellectual property strategy. So those two aspects will be very important if we want to continue our collaboration with industry partners in the future.
Thank you so much, Mateusz, for this fantastic overview of your project, the Drug-Repurposing Project, and also addressing all these important areas from starting from all research, from old medications used in one therapeutic area, and then basically discovering its potential, their potential in oncology, through all these areas that you cover related to technology, to creating the combination, adequate combination, and at the end of the day, preclinical and clinical data through, again, regulatory science tools that can accelerate these combinations to come to the patient. Thank you so much for your time and for presentation of your fantastic project. Thank you.